Okay, welcome to the Open Source Antibiotics Mur Ligase meeting. This is Tuesday, 6th of July at 2 p.m. Thanks for coming along. And the agenda and actions were posted last night, and I will just bring those up so that we're all looking at the same thing in case people don't have it in front of them. And that is going to be hopefully that page. So there are a bunch of things here which are sort of sitting here as to do items, but I guess there were a few things that I just wanted to try and cover first. So just on the shipment of the atomized compounds, which we're all very excited to test. So we do have this approval now to ship to Diamond, but we were holding out for agreement to ship directly to Warwick first, EULAR, I think, so that you could have the compounds and then maybe take them to Diamond. And we are, I think, very close to getting that. The document is back with UCL's legal team, and I'm hoping that they can just agree to Warwick's queries and questions, and then we can get going. In the meantime, Dana's taken a look at some of the compounds, a sample of the compounds, and they look fine. So we don't have any issues to do with things going off because they've been kept properly. So I think we're still fine. And I was going to give the UCL team just a day or so, and then if there's any issues with the remaining paperwork, I was gonna maybe try and think of a way around this, like maybe shipping to Diamond first, even though that's like the plan B. But certainly in the short term, I think we'll resolve this one way or the other. That's where we currently stand. Nothing really for anyone to do there. It's just that I'm keen to sort of get this going. In part because we've had just a new input in the last few days from Jan, who's with us, who's been working on docking some of the some commercially available compounds into Mur. So I was hoping we could just maybe start with this because it's kind of relevant in the sense we're again talking about AMID, commercially available compounds, which appear to be scoring well. So you posted everything here, Jan. I mean, did you want to talk about it a little bit and just bring everyone up to speed in case they haven't seen what's been going on here?
Yes. So yeah. So sort of. Sorry.
So do you want me to come off the share and you share something, or shall I just scroll?
No. I just wanted to sort of. It's basically more where we're coming from. So Casper Steinmann and I wrote a paper a couple of months ago. Where we use genetic algorithms basically to create new molecules that have good docking scores. And so as part of that, we also compare the docking scores to commercial databases, just to make the point that we can actually get significantly better docking scores. And so that's typically the first thing we do, so we sort of know what docking scores to beat. And so we've done that. And I just thought I would put those up. So the genetic algorithm is running right now, and we'll get other molecules that have significantly better docking scores, but of course won't be commercially available unless it sort of gets lucky. You can see a question here.
Oh yeah, thanks, Hans. It's Chris Dowson, Jan, from Warwick University. It was great that you're engaging with this. Brilliant. I'm just wondering, as part of your algorithms, whether you can put into this it's something like the entryway program that's used to understand getting drugs into the gram negatives as well. I see a lot of heterocycles. I'm just wondering, the relative hydrophobicity of these.
So I mean, one thing we did to check was just to log p-values. So if you go into the notebook, you can sort of put your mouse over each molecule and you get the log p-value, and they all seemed reasonable. Okay. In principle, anything that we can predict relatively quickly, we can include in the genetic algorithm. So we can automatically throw out molecules that have a high log p-value, or basically anything that we can compute from the SMILE strings.
Yeah, Chris was referring to a set of sort of heuristics about what makes molecules accumulate in gram-negative bacteria. So features like primary amines and things like that, and whether you can apply and globularity, right? So things that work best. If there's a way of applying a set of rules on top.
Yeah. So we can do that if we have access to the software package, basically. So if someone can put details up on the issue there on the channel. But I assume this is something, well, I should ask, is this something that's reasonably quick to compute from the smile strings, or is this something?
Yeah, very easy.
Okay. Yeah, it's just sort of in the course of a genetic algorithm run, we screen about 400,000. So it has to be fairly quick. So basically what I just need, is it a commercial piece of software? Is it something we can download?
It is a web portal set up by a research group, which may not do very well with large batches, actually, come to think about it. I think some people on this call have maybe tried that, and we realized that we have to put a molecule at a time. Is that right? Has anyone tried it?
Okay.
Yeah, I've run it before. I think you can input batches, but it takes some time to get back to you, I think, if you do that.
Yeah. So that's a problem for us for doing it on the fly. So basically, we try not to do anything where you have to use the network, just because for all sorts of reasons. So there are two options. One is to do a post screen, or a pre-screen, actually. If it turns out that I mean, if you're doing it for a database, right? We can, well. Yeah, so let me back up. If it's something where you have to sort of, where you can run natively, where you have to call and rely on network traffic, then we can't. It's for practical reasons. It's hard to do on the fly.
I wonder whether it's worth a discussion with Paul Hergenrother's group in Illinois, who wrote that entryway program, and maybe he might prepare to open up the code and maybe think about being able to integrate it as a single package rather than having to,
that's a good idea.
Internet. And I think he might be the type of person who might be keen on that. That'd be a good start point.
Yeah. No, I can definitely look into it. I mean, sort of if we can get the number quickly, then it's not a problem to include in the genetic algorithm. So we already have things in the genetic algorithm that screens for synthetic accessibility and things like that. And we also almost put in the log P, but it just turned out that we get molecules with reasonable log P values anyway. But we could also throw out high log P values if we want.
I mean, obviously, the thing that caught my eye was that the top couple of rows here, the zinc hits, are scoring better than our AZ compound that we know crystallizes with Mur-C. Now, of course, you can't read into absolute values, but that's not a bad result. And so, to my mind, what Chris said is totally relevant and important. But if we could access a few of these and check whether or not they actually bind, then that would be an amazing result, because it would bring up a bunch of structures that we weren't looking at before.
Yeah. So it's so I did a little check now on the actual availability. So basically, we weren't very thoughtful about picking we picked the enamine because we knew those were purchasable. And the list of zinc compounds we just had lying around from other stuff. So these are not necessarily in stock. Before the meeting, I just checked the top 10 with this Mopart program that was suggested, and only number 2 and 4 are actually in stock from enamine, I could see. So I can check the other ones too. So I mean, if these are really, so I guess with the libraries, there are several options. I mean, it's possible to just use in-stock molecules from zinc. And I'm trying to download that set of compounds. And also I think the reason that the zinc molecules score better than the enamine is we used a small enamine set of only 10,000 and a large zinc set of 250,000. But there is an enamine set of 250,000. So we could also try to dock that instead. So I think in principle, as far as I know, the enamine set should be all purchasable. Maybe not in stock, but maybe sometimes they make them on demand.
But the top zinc hits, they're not difficult to make, right? I mean, they look single steps if you're lucky.
Okay.
I would guess. Just look. I mean,
yeah, that's the other thing we can do is so we can interface this with the retrosynthesis program and basically give you for each molecule the minimum number of synthetic steps for each. So that's something I can add fairly easily to the list, and then we can rank them based on that too.
I mean, that would help. Card carrying chemists, we should be able to do that pretty quickly. It feels like using cruise control. It's cheating. But
but if you're doing a lot of them, right, then it's But then I think it might be worth waiting for the genetic algorithm ones, because they will definitely have a higher docking score. I mean, if you have to synthesize anyway, they will definitely have a higher docking score and maybe will be equally easy to make.
Okay.
So I think those will come next week sometime.
Oh, wow.
They're running right now.
Okay, that would be fantastic. Yeah. Okay, let's do that.
Yeah, so also, I just from my point of view, I mean, it would. Of course, we want to help in the effort, but we're basically where we're coming from is if we could get some experimental validation on the genetic algorithm predictions, right? That'd be wonderful for us.
Yeah, absolutely. Yeah, completely understood. And that's what we want to.
Yeah, and that could also be a paper. I mean, one paper just on more on the experimental validation of the method to sort of show that it's worth spending the extra effort with the genetic algorithm to get even higher scoring molecules.
Yeah, no, awesome. So, I mean, just on the, I guess there are two follow-up questions. One is that I assume that how these molecules are being predicted to bind, so the pose in the enzyme, is that right?
Yeah.
Okay, and so I'm assuming that if we could cluster them, we might learn something about the value of certain residues as binding partners for small molecules.
Yeah.
Is it possible to generate that?
Yes. So the person who's actually doing the docking is my collaborator, Casper. He's on vacation this week.
Okay.
But I mean, I don't know if he saved them because we did 400,000 per run.
Right.
So, but if not, then we could certainly do let's say the top 1,000 ones again to just redock them and then this time save the pose. Okay,
great. I mean, I guess I'm kind of interested in the 3D shape of what they make as the ensemble, And then the second thing was, so a lot of this project is based on the idea of one molecule hitting more than one enzyme, a so-called multi-targeting approach. Yeah. So I was wondering if it's possible with the results of the genetic algorithm when it's finished to run, the sort of top 100 against one of the other Mur Ligase enzymes. Chris, I'm assuming that would be a good idea, or if we could pick something as similar as possible and see if it's also predicted to bind there.
That would be perfect.
Yeah, we could certainly do that as long as we have the X-rays or any structure.
Yeah.
Then we could also, I mean, I think that the redocking of the top 100 or whatever is easiest, but if that turns out to be sort of a promising direction, it's also possible, in principle at least, to dock both doing the genetic algorithm and sort of steer the search towards, let's say, binding one but not the other, or binding both, or whatever you're most
really
interested in.
That latter sounds really attractive, actually. If you have a level of integration right up front across a pair of ligases. It depends really on how much computing power, but in an ideal world, you'd be looking for a pair of ligases and then picking off several species simultaneously that could form a clinical indication that would be your target product profile. You then got, you can go for urinary tract infections or respiratory infections, and you're immediately focusing your screen to give you the best hit against the 2 or 3 pathogens that are the most important, and multiply targeting. So small ask, but that would be lovely.
Yeah, I should just throw out the caveat, right, that this is docking. I mean, I'm sure you all know this,
Understood.
but
Understood.
How good it is.
We know. I have a colleague at Warwick who rather demeans it and calls it guessing with style. But
I think it's very target specific, actually. Some targets that the scoring function, at least in Glide, which is what we're using, seems to be picking up the physics, and then some targets that just doesn't. We tried something similar with the COVID main protease, where they then generated a lot of data, and there is absolutely no correlation between the docking score and the IC50s on that system. So, yeah.
Right.
That's brilliant.
Peter had a question.
Yeah, I was going to ask about I see a number of compounds that are very similar looking to me with my naive eye. Is there a way to sort of figure out what are the fragments that are similar in a lot of them, and then see if you can just bind that one, so that we are buying maybe fewer but more likely hits that rather than so many, and maybe there are multiple ones of the similar sort of warhead?
I can certainly, I mean, what's very easy to do is just calculate a similarity score sort of between them. So and then sort of set a cutoff. I think the usual sort of cutoff in drug discovery is, I don't know, either 0.6 or 0.8, you know, where 1 is a perfect match. So we could sort of indicate if there are other higher scoring ones. So we could basically, we could eliminate lower scoring ones that are very similar to higher scoring ones.
Yeah, that makes sense. I mean, I'm just saying that like there are many of them, numerous ones here, like the top left two are very similar. The warhead is similar. The sort of the linkages are different, but
Yeah.
I just see like a number of them. Yeah, the ones that your cursor's on, those two are also, yeah. So I just think that, you know.
Yeah.
Yeah.
The question is, and we can certainly do that. I should say I'm not really a med chemist. So the question is, at what point will I make, what I'm worried about is sort of making stupid decisions and you never seeing the molecules. So at what point sort of do the experts come in and just sort of, you know, throw them out? And at what point do I do it?
No, I think Peter's Peter's worried about, you know, doing crystallography, let's say, on some of these, and instead of doing 15, you want to pick 5. Do you want to, just because the experiments are quite labor intensive, and so he's thinking, well, let's try 5 diverse compounds.
But then it's probably best that like an expert picks them. I mean,
no, I understand. I agree with but I was just sort of making the same point. I think that what Matt was making is you could have the structures, you could sort of see what is binding and just keep the piece that is binding instead of all the sort of extra stuff that seems to come along
so
as a gift.
So like a tree, right, where you cluster similar structures.
Okay.
You think, okay, I'm going to pick one from each branch, you know.
Yeah. Exactly.
I can look into how to make that and try to make that. It sort of depends. I mean, if I guess you know, if you have to sift through 1,000, then it's probably best that I code something up. If you have to sift through 100 to pick 5, I would probably just eyeball it. I mean, it's just safer.
You can do that.
Yeah.
Okay, fantastic. Okay, well, we'll wait the outcome of the final search that's going on there, and then we'll make some decisions about whether to make or buy and what we're going to do. But this is an amazing start and very exciting, because of course it's a 4th strand to what we have here. It's a bunch of new molecules that we could get access to and start evaluating.
Yep, and at some point when there's enough data for machine learning, then we could also, I'd be happy to make some machine learning models, basically trained against IC50s or some experimental data.
Okay.
That might be more reliable than the genetic algorithm. Just
that's great. And Matt, I'm just thinking about your tree, not just for these compounds to look at their chemical relatedness, but for all of the different compounds across all of the different start points we have. I'm just, you know, thinking, you know, I feel a bit Paddington Bear. My brain is too small to hold all these things in. You know, we need, you know, a graphic that we could assemble and display all of these, and particularly when we start getting synthetic derivatives from them, so we can have some visualization as to what we're talking about rather than just code numbers. I don't know if that's you know, possible, doable, easy, difficult, resource intensive.
It sort of depends. If someone, like I said, it's not my field, but if someone knows of a package, I can also Google around and see what I can find. I can talk to, probably Chris Swain would be a good person to talk to about this. And if it's something open source that we can just, you know, that's easy to download, then we can certainly try it. So I just, if you if you guys give me input or suggestions, then I can try it out. I don't think I have the background to come up with my own ideas, but I know RDKit pretty well. So
for visualization, we usually use Data Warrior, right?
Okay.
So if we've got a spreadsheet of molecules and we want to see them, then we use that, which generates something that's not unlike what's on the screen here. But
yeah, I should also say, if you go to the notebook, maybe I'll just try to share that. Yeah, I think I'll try to, yeah. And then I just want to go in and show the notebook. So it is, just have to put it down here. You can see it now, right? So it is somewhat interactive. So you can see I can like put data on top, right? And they're also, these are just the first 20, right? But one can scroll through, one can also search or sort by anything. So if any information that I can quickly calculate, I can easily put in, and one can sort by it. One can also go in and select molecules, for further study. So there is a little bit of a user interface here, but if I can also, I think Data Warrior, if that takes a CSV file, then I can also sort of make one with the appropriate data and make it available. So yeah, just give me, we take requests basically.
Yes, SDF will be appreciated if you could.
To put what?
SDF.
SDF, sorry.
Oh, SDF. Oh, sure. I mean, just a big SDF file with everything.
Yeah.
Okay.
And just one small question. About this project? How many compounds are we gonna screen actually for biologically? So we can get enough data for the machine learning generation because the generic prediction will be validated only if we could get enough data to measure the success rate, like what kind of predictions can be useful, what kind of predictions are not.
That's a resources question, right?
Yeah.
As many as we can.
Yeah, I mean, for machine learning, sort of I would say the absolute minimum is maybe 50, and that's really dicey, right? 100 is a little better. But sort of as a validation for the docking, I mean, I guess it depends how many. I mean, if the first one is a hit, that's right. That, I mean, there's other things obviously that needs to be optimized. But if our top molecule has a good IC50, then I'm happy. So,
because you were talking about like using selecting compounds based on their predicted docking score and log P, I think it's log D, right? Log P, yeah. So I was wondering, like, do you do the visual selection at the end, or do you just select all by their scores?
Okay, so in this particular case, it's just the docking score.
Just docking score.
And also there is some synthetic accessibility. Well, actually not in this one, since these are purchasable, but in the genetic algorithm, there would be synthetic accessibility. For the purchasable ones here, it's just plain old docking score, because we just sort of by visual inspection, it looked like the log P was fine. I believe the rule of thumb is below, I don't know, 3.5 or 4 or something.
I saw you published on the GitHub saying that you were using the Glide SP for the crude virtual screen, and then you go for the XP for more accurate predictions.
Yes.
And eventually select. Okay.
Yeah. So we take the top 1,000.
Do you think we should try the Chris suggestions, like trying different algorithms to score the same data sets, like I mean, from the scratch instead of just, rescore the 1000, because if we screen from the scratch, then we can see what's actually overlapping in between. And those might probably be, I don't know if it's quite nice, but yeah, I would say I would take that as a bit more preferable source of data.
Yeah, that's sort of known as a consensus score, right? So
yeah.
that's used a lot in machine learning also in general. That the main sort of practical issue there is just that, I mean, we have experience with Glide. We have some experience with something called SMINA, which is like AutoDoc.
Yeah.
But we're not experts, and it's just hard to sort of reach the level of proficiency required to use the docking properly for a bunch of different programs. So it would be better if we could gather some help from other people like David Coase, who made the SMINA and AutoDoc Mina and things like that. If he would do the docking, because then it's done right.
Yeah, I've been thinking because UCL has like HPC resources, we can definitely accelerate the docking process if we can make use of it, like the Thomas or the Catalina, that they're all available.
Okay.
So I mean, if we can have some software like shared on this platform, so we can just so we can do a lot of things cooperatively.
So I mean, all the basically all the smiles strings which you need, or we can also give structures. I mean, these are just, I mean, the both enamine and both data sets are basically publicly available. With the genetic algorithm, of course, it would have to be hooked up to the genetic algorithm, but maybe just a rescoring. So running genetic algorithm with one algorithm, docking algorithm, and then rescoring it afterwards with other algorithms would also be fine. It's just, I think the main, like I said, the main limitation is just experience with the docking software. That can take a long time to sort of become an expert at and not make stupid mistakes. So I think that would actually be the rate limiting step. So I think it would be better if we could get people who are sort of experts in the various docking packages involved, and they could get our compound set, and maybe they could give us some compound sets that we could compare.
I think that sounds very good. I think we can take this up on the issue that was started, but that sounds very good because, of course, as you said, we could just rescore things fairly easily. We can find people willing to help us out.
Yeah.
Great. Peter, did you have your hand up for historically, or is there another question?
It's a sticky hand, sorry about that.
Okay, no worries. All right, that's fantastic. I just want to move on so we can get through a couple of other things, but that is a really great input, and we'll pursue this after the meeting. Okay, let me just stick this back up, which is here. Okay, so I guess the other thing I was hoping to talk about is so the stuff that Becca was doing with both the fragment screen, where some of the data have been put up on the website, which is great. Chris wanted the structures of the molecules to be added. I'm not actually sure if we have that. I'm assuming we have the structures of those fragments somewhere, do we? The ones that were evaluated?
I don't know if Lizbe does from her side.
Yeah, I do have the structures somewhere.
If they could just be dumped somewhere. If it's a big file, then just a sheet, but if it's a small file, then structures or something, that'd be great.
Yes.
Okay. And then, as I understand it, there's been some progress on the evaluation of Dana's fragments. Did Becca and Dana, did you want to talk about these? Because I think it's pretty hot off the press.
Yeah, so we did the initial screen with all of them against Mur D. Got a few quite good hits. We got about 11, which reduced activity by about 80% or more. So we double checked they were true inhibitors by testing against the coupling reaction. And then true inhibitors, we tested against Mur E from the same bacteria, and we appear to have a couple which do seem to be dual inhibiting, which is really good.
No way. That's the biochemical inhibition.
Yeah.
Wow. That's amazing. Do we know what they look like?
That would be on Dana to tell you what they look like. I worked off of just numbers.
Okay.
I mean, I can share screen if we want to do that now, or can post it later.
I guess, well, yeah, why not? Come on, we're live, why not? I mean, I guess my question really about the 11 hits is whether they clustered structurally, you know.
Oh, so I didn't look at all 11, but these are the 6 that were tested against Mur E as well as Mur D. So yeah, they actually, I don't know, all the fragments that we sent to Becca were fairly similar, but some of this sort of extending off the phenyl ring, it looks like.
What's the concentration here, and what is the enzyme? The specific enzyme.
Becca?
So the concentration was 1 millimolar, and they're both from strep A galacticae.
So sorry, wait, I'm just noticing what the y-axis is. So we're looking for inhibition is way, we want them way down, right?
Yeah.
So the most promising compounds are what, 742, 754 or something?
Yeah, so it depends on how we're grading, how we want it. Obviously, some have higher against one, but are more equal across both. So depends how we want to move forward in grading these. So the 754 is very active against Mur D, but has slightly higher against Mur E. So,
I mean, these are high concentration fragments, aren't they?
Yeah.
What was the concentration of the enzyme?
The concentration of the enzyme was 2.5 micrograms per mil.
Do you know what that is, millimolar?
Not off the top of my head. I can work it out.
Because it's that molar
Yeah.
ratio between the enzyme and the inhibitor. So
How many compounds were evaluated?
78, somewhere around that, I think.
Right. So it's a small number of those are hitting. So it's not as if everything's active.
No, there was definitely some which definitely were not active, some increased activity within the assay. So
I'm just scanning through to see whether you had some examples of them something that was really potent, a potent single targeter, or whether you found that when they targeted one, they targeted both.
I'd say I think it was 749 was mostly only targeting Mur D. Against Mur E, there was not a level of inhibition we would be expecting.
So okay, so these are all the one. This is everything that showed some level of inhibition. So
yeah, I didn't rerun the entire 78 against Mur E yet.
Oh, I see. Okay.
It was just ones which were inhibiting Mur D that we went and then tested against Mur E.
I understood. Okay. Very good.
Yeah, I'm liking that N-methylpiperidine. Wow, that's amazing. That's on a bunch of actives there. Okay, we're going to need to digest those. I mean, it's going to be important to compare the actives and the inactives, right? So if we find there is a cluster there with activity and there are the same motif is not found in the inactives, then we are all guns blazing for that. Looks nice. That's really good.
I was wondering whether we kind of did the retro with Jan, whether we sent those compounds for him to dock and just to as a benchmark, see what docking scores he would get for those, just as a bit of a. I think we've lost him now.
Amazing.
Yeah.
Oh,
Yeah.
hi. Oh, yeah. Sorry, I can't. Oh, you're off on screen. Yes, hi.
Just from experience, I mean, small fragments will have low docking scores. It's just, yeah, I can say that right now. But I mean, there will be sort of a spread in the docking scores that might be. It's easier for us to do if we get this. We just need the smile strings or the SDF file.
Do you want to?
So is there a plan?
Is that worth doing?
Is the what? Sorry, Chris. What did you say? Sorry.
Is it worth even thinking of just getting docking data, just so we having full sets of data for all the molecules that try and because we'll be doing the reverse, won't we? We'll be taking the best docking scores when we're doing the biochemical assays. I don't know.
I'm happy to do it.
Okay, awesome. I mean, absolutely. I think that would be amazing. I'm guessing the other strand to that is that the ones that are looking the best here, we need to try to get them crystallized, right? I mean, is that the next bit?
Yeah.
Yeah, I was gonna ask about that. So maybe we should
Can
give a try. Can you hear me?
Yeah.
Yeah. Sorry. Maybe we can give a try for co-crystallization with the top compounds. Something like that. It takes a while until we get the Mur crystals. That's like between 1 and 2 months. It's a very slow crystal system. So I can throw more than, maybe like at least 5 compounds so we can have something. For Mur E is faster. So that can be another option that, yeah.
Laura, I'm just thinking because of the speed of the crystal system, when we're trying to do things iteratively with no money, it would be really handy if we had a better crystal system.
Yeah, that's what's gonna be one of my comments after my update.
Okay,
for Mur D, I think. So I've been trying to get a new crystal system for Mur D, and it's not going that well. The crystals are too small. They're not very robust in growing. So they grow a bit faster, but they're not consistently growing. So I don't get like a lot of reproducibility on the plate. Which means it's not feasible for XChem or high throughput screening for crystals. So I was thinking that maybe we can consider another organism for Mur D, and I can give it a try with that and see if we can get something faster and more reproducible. Because waiting that long for getting a crystal is gonna be a bottleneck for the project. I don't know what you think, but yeah. So I was using the system done by Dom from Streptococcus agalactiae Mur D and E. coli Mur E. That's what we have here.
Joe, you were trying to come in there when we started talking about crystallography? Did you want to say something about the plan?
Yeah, I just want to go back. I mean, I think it's important to actually do the dose response for the compounds that showed some inhibition, and to actually look at the hill slopes and the curves of these compounds. I think it's very important that those experiments get done rather than just looking at percent inhibition at this point.
Yep.
And obviously, again, if ultimately, if you want to do anything about a little bit of SAR or something like that, the percent inhibition is not of that. It's interesting. It highlights which compounds maybe to further investigate. But until you get really some dose response and actually looking at I would say looking at the hill slopes and how these compounds are actually behaving biochemically. I mean, that's the enzymology that needs to get done.
Is that feasible?
Yeah, if we're just gonna target the ones which we think as dual inhibition, then yeah, those slopes are definitely fine. Obviously, if we're trying to do all 78 against both enzymes, it would take quite a while.
There's no reason to do all 78. I think it's if you're just if the goal of the team is to look at dual inhibition, just looking at the ones that are dual inhibitors and really profiling those to see. Again, I'm just very, these fragments in this weak activity, you don't want to be chasing ghosts here.
Yeah.
And also in terms of competition, I mean, I guess this is in the level, this is a competition assay. I mean, but you're not actually, in this case, you're actually not even competing with ATP. As I recall, these are binding into quote an allosteric binding site and not the actual, they're not ATP competitive, as I recall.
Dana, is that right? Are these the allosteric ones? I forget the cluster it came from.
These would either be the ATP or the substrate binding site. And let me just check because it'll be on.
These are all. I thought these were all site 3, but maybe I misunderstood what you were doing there.
No, I forget that we got clusters in various places and went after several of them. And so I thought this was allosteric.
Oh, yes, Joe. Sorry, you're right. Yeah, remote binding site.
Right.
Right. It's definitely convinced me we need a picture for us with small brains.
Yeah, it's on the website. But again, I think allosteric inhibition is obviously, is kind of in vogue in some disease areas right now. But again, I think as was pointed out, you don't necessarily, I guess these are functional. Is that correct? In the sense that in your assay, you're looking at turnover of substrate. Is that correct?
We analyze, yes, we analyze the amount of free phosphate being formed from the conversion of ATP to ADP.
So in that sense, these are functionally active.
Mm-hmm. Yep.
Yeah, it's not just binding. I mean, it's not binding, it's functional, yeah.
Okay, yeah, I've just got the structure up here. But anyway, so the fragments are in green, and then the purple is the substrate site. So up here. So I thought that might be helpful, but yeah, it's on GitHub.
All right, great. So the action there is to look at the dose response and maybe to see if we can figure out if they're being bound in the same place. If we know the fragment that these come from, we could, of course, try to see if they're binding in the same place. I'm assuming they would be, but you never know. And to do a kind of cluster analysis of the actives versus inactives. Those are all the actions on us. Nice data, though.
Well done, Becca, team, Dana. Excellent job.
Yeah, that's great.
Okay, with the hill slope, I'll go and talk to Adrian, but do we want a full 10 point data set for each of these, or can we get, what would you be happy for us to get away with concentration wise?
Well, I mean, I think, again, I'm not an enzymologist, and I don't play one on TV either, but I mean, I think typically if you want a true hill slope, you're not going to do like a 4-point data set. I mean, you're going to do a 10-point curve to get a true accurate hill slope. Now, my understanding is in hill slopes that basically once you get above 1, if you get like a 2, then it tells you something about cooperativity and so on. So again, I guess it's it's interesting that these are functional in that sense, that you're inhibiting the actual reaction and non-competitive, and maybe that's, I guess the question there becomes, when you start looking at that binding site, how conserved is it across the relevant, for example, escape pathogens, right? I mean, one of the things right now you're looking at, like for example, Mur D. In Mur D, you're looking at that galactate or whatever it's called. I mean, you're not looking at E. coli. And so then what's the actual, once you start getting away from the binding site, what's the conservation? What's the conservation in that site 3 that's allosteric? So I mean, those are kind of questions I would be asking at this point in terms of pursuing that binding site and getting a little bit better understanding of, if you're dual targeting, because that's going to, if you go to a grain application, those are the kind of questions the reviewer is going to be asking, right? They're going to be asking, okay, what you're dual targeting, they want to see, the sequence, they want to see the structure, they want to see that, that there's, a logical path forward in terms of dual inhibition of relevant clinical pathogens. So, yeah.
Great. Yeah, true. Okay, just conscious of time. We can't run through everything here. I just wanted to ask if there's anyone who wanted to provide an update on any of the stuff that's on the screen that hasn't had a chance to talk yet. So open mic.
Yeah, I guess I should say there's an action item for me there about going through the literature. I sort of that they came up from making a machine learning model. I think the bottom line is, if I get the data, I'm happy to do it. I'm not going to be able to find the time to go trawl through the literature. Also, just because I'm not really equipped for it.
Sure.
So just, yeah.
I sorry, I got caught in a call. My mother phoned. Yeah, I mean, rather than going to hill slopes with all of those hits, and I think my temptation, Joe, is to follow up on your suggestion around priority pathogens and see if any of those hit in D&E of other pathogens as well. And then if some of those hit, then they in my mind, they'd be the ones to focus on. Because you're right, we could end up with, you know, some nice inhibitors of group C streptococcus, and no one's going to be interested in funding that.
So what's the status with actually Mur C in your lab? Because I think you said way back in the day you did LifeArc screen, with, I think with Pseudomonas maybe. So have you gotten that out? I mean, that's all. I mean, that's, you guys are driving this, you know, project and want to make funding in the UK, but so, you know, you guys can decide which pathogens. But again, I think trying to zero in on, especially like, again, for E. coli, I would be thinking, I mean, from Mur D, I'd be thinking about E. coli at least. And for Pseudomonas, I would, you know, from Mur C, I would be thinking about Pseudomonas and getting those assays up and going. And ultimately, that's the data I think you'd want to present in the grant application.
Yeah, I agree, Joe. Part of this is legacy stuff. Having had the Agalactiae XChem, that was the only crystal that made the XChem cut. So, you know, that's why that was the XChem that ended up getting driven through XChem. So it's part of it is historical, and likewise then designing around that.
Okay. Yeah, like I said, I don't really want to go through everything here, and I hear your comments earlier, Jan. I mean, I guess we've got a new thread to everything now, so I might just leave that there for the moment. But if it gets really mothballed, then we can take it out. And I completely understood your comment about bandwidth. I think there's nothing else here that we need to do.
I guess, Matt, I'm sorry, you solicited questions or comments.
Yeah.
It's great to see the update on the chemistry.
Yeah.
And 55 mgs of looks like one of the compounds.
I think so.
So pardon?
We're just waiting on the NMR. We're kind of biting nails, waiting for the NMR.
Right. So is that going to be something that you can share with Peter, for example?
Yeah, immediately. As soon as we have confirmed the NMR, it's going in the mail.
Okay. So I don't know if you have Peter. Peter, you guys shared your shipping address, or I guess it goes to Bart first or something like that. But the logistics of that, I guess, is an action item of how to send that compound on.
Yeah,
because that'd be fantastic if we can get something in, you know, that compound. I guess we need that in ideally E. coli or Acinetobacter, I guess.
Yeah, I think you need to go to Bart first, and then we'd be happy to get it. I think it needs to go to Bart first, and then being happy to get it.
Right. But I think the ones that the isozymes you have are, I think, in terms of Mur D, what do you have? You have, I can't remember. You have the Acinetobacter.
Yeah, Acinetobacter and Pseudomonas are the two that we have.
Yeah, so that's great. I mean, if we could, if they could, you know, if you can get a structure of that compound in one of those Mur Ds, then that would really, I think, really form a foundation of a grant application for you guys, for Matt and the UK folks.
Okay, you know what? I'm just gonna, when we have this thing confirmed and when we're figuring out what we're going to do in terms of where we're going to send it and why, I'm going to CC you in, Joe, to make sure I get my Latin terms right and we're talking about the right enzymes. Okay, so to make sure we've got a consensus on what we're going to target it against. The Yuhang's then subsequent targets will be one of the original compounds with the methyl up on the top, and also, you know, I mean, this compound 9, where the OH has become an amine. I mean, that still remains one of the targets, right? Because modeling suggests
it's compound 4 actually in the paper. I don't know where the compound 9 came from. It's compound 4 in the paper.
Oh, that's probably Yuhang's local thing. Yeah, that's an internal thing. I think you're gonna have to start using your own codes, Yuhang, because it's going to confuse people. So yeah, that's just in the synthetic scheme because we're going from 1 through to the end. one question we had, we were talking about this, and we wondered if there's any use in having a negative control, so a compound from this series that doesn't have any activity. Is that useful, or would that be not useful? Control compound, for biochemical.
I don't really see that. I don't see that as of high value personally, but that's my opinion. I don't know, others may differ.
I mean, like in a biochemical assay, like in a biochemical assay, if you want a compound that looks like that but is negative, is that helpful?
I guess I would say if we ever get to machine learning, it's super helpful.
Right. Yeah, that's true. Okay.
So I guess the only comment on that would be is if you, well, first of all, you don't even have the assays to say cross-reference with the AZ paper, right? So the AZ was doing Pseudomonas murci. So sorry, Chris, but until Chris's lab has Pseudomonas murci biochemical assay up and running, and once you had that, then you could do basically, a couple of data points, and then you could use all the AZ published, activity, which I think there's probably some compounds in there that are not as active. So you could use that as your trainings.
Yeah.
You make that accessible, and you can combine those two data sets, whether it's data out of Chris's lab or the AZ paper.
Right.
But anyway, it's again, the critical step here is going to be at some point, you've got to get a biochemical assay. I mean, because right now, Chris's lab, you said you have the galactide, Mur D, and then I guess you got to pull out of the freezer your Pseudomonas murci and kind of get it going again.
Yep, fair enough.
You're in mute, Chris.
Thanks. Yeah, I'll have a look, Joe. I think we may well use it frequently when we're making reagents, when we make the intermediates. I'll have a talk to Adrian. He's just been going through all that. But I don't bump into him very often.
Thanks, Chris.
Looks nicely crystalline there, actually. You might get a crystal from that. Yeah. Look at that.
Yeah, I will make more of that.
Great. Okay. All right, great. I think that was everything I had. And we're 5-2. If anyone's got anything urgent, please say now. Okay.
I did send things to AlphaFold, so that's a tick done. So
great.
That's with them.
Okay, and the ELF situation, we're in discussion still?
In discussion, they're wanting data. So what they're wanting is to have already run the high throughput assay. So not the assay Becker's been doing, but the next generation, which is very low volume fluorescent format. So what I have to balance is keeping ELF happy and doing that, or keeping Joe happy and doing Mur C. So it's a very, I'm torn.
Well, you know what my priorities are, Chris.
I know, Joe.
Just please bear me for one last question. I think I've been searching for literatures for quite a while about that. Like it's about the Mur C and Mur D, those crystals, the crystals of the protein. They have about the crystallography of those. I saw them having like manganese or magnesium metals in the center. I'm not quite sure if they were like originally in it when we found the Mur C, for example, in the bacteria, or do we just add those metal ions in? So that might influence the outcome of docking, because like when we are actually doing the docking, we consider about the metallic interactions, or that depends. So if that metal is not involved in the biological process, then we probably might need to just delete that metal center and just consider all about like
So when we're doing docking and crystallography, the question is, you know, what else do we have in there by default? I guess that's a question we've always been wondering, right, Chris? Anybody else?
Yeah, Laura, we have the cations in there, don't we, when we do the crystallography?
So when I'm setting a crystal place, I'm adding magnesium, but I know the initial screens were not done with magnesium.
And when we do,
that's why I wanted to redo the Ferrins screening in the presence of the magnesium.
Yeah, and Becca, you've got magnesium in your assays, haven't you?
Yeah, if not, it won't work, isn't it?
So do we need to count the interactions between the metal and the ligand then? Because those were added.
Yes.
Yes, they need to. They're going to be on the cell. Yeah, they need to.
Yeah. I mean, if they're obviously if the ligands near to where the metal binds, obviously, but yeah.
So I assume the protein just took those metal ions from the blood anyway. So they will have similar binding situation as well.
The metal ions will be in the cytoplasm of the bacteria.
Yeah, that's what I'm thinking. Oh, thanks.
Sorry, Lizbe, were you trying to say something there? Because I'm sorry you were drowned out.
I know, I just said magnesium is normally needed for ATP binding, so inactivity.
Agreed. And you can't tell with crystallography.
But it's going to change the atmosphere in the area. So yeah.
Yeah, I agree.
Environment is going to change, though. Yeah.
Yeah, but you can't tell with crystallography because it's the same number of electrons as like a water molecule, and so you can't. There's no way that the magnesium has no anomalous signal, so we can't tell whether it's water or magnesium. But you can only tell on the coordination.
Right. But the structures that we're using for example, the modeling that Jan's using, the expectation would be that the metal ion is in there. Is that what you mean?
Sure. I mean, we can't tell what's water and what's magnesium is what I'm trying to get at. You can only tell from the environment. So if it's coordinated as a magnesium and it's a magnesium, if it's coordinated as a water, it's a water. We can't tell the difference between the two. There's just no physical way of telling crystallography of whether something is a magnesium or a water. Every other element, you should be more lucky because they may have an anomalous signal, but magnesium doesn't.
Okay, great.
So it's just all the docking I did back in 2 years ago. I mean, we get rid of all the waters and metals and stuff when we were doing the docking. I don't know what Jan's done, but typically we eliminate the waters and any metal ions and so on.
I think we do the same. I'd have to just confer with Casper, but I'm pretty sure, yeah, that those are removed just because they're, it's uncertain what they are, and they're very labile, right? So maybe one ligand will throw it out, another one will need it, and yeah, so you just end up throwing it out everywhere for every ligand. I mean,
so just confirm, like we delete metal and water.
Yeah, typically. In the
docking. Yes.
In the docking.
Yes.
Okay, right.
And just a comment. I mean, there's so much work done in kinases, which are obviously ATP dependent, and all that stuff, and all these approved drugs, and they're all mimicking, or displacing metals and the phosphates and all that kind of stuff. So I mean, it's all around enzyme concentrations and in terms of IC50s and all that kind of stuff. But just in terms of docking, yes, those are always taken out of any of these kind of docking simulations.
Okay. Right. Thank you so much.
Okay, great. I'm going to have to run. The next meeting will technically be beginning of August. People will probably be on holiday. It would be nice to catch up if people are available and no pressure. I might just put it in the diary if that's all right. And it's either the 3rd or the 10th of August. Don't mind. Any immediate preferences? Okay, I might go for the 10th in that case. And like I said, if you're on holiday, don't worry. All right, great to see everybody. Thanks for coming along, and lots to do. See you next time.
Okay.
Bye.